Сегодня спикерами конгресса стали действительно именитые люди, и рад представить вам следующего спикера, руководитель научной группы Исследовательского института перспективных исследований ICREA, Барселона, Испания, Давид Торрентс Ареналес. Друзья, ваши аплодисменты.
Hello everybody. Can you hear me? And some people have translator. Okay. Hi, I am David. I'm coming from Barcelona. I am not a medical doctor. I am not a computer scientist. I'm something in between, which there is a big area. I will try to explain you today things that are happening in the world or in the field of genomics, and mostly genomics applied to the clinics or to the patient directly. So first of all, I did my PhD on molecular biology in the laboratory with a pipette, very close to the medical doctors. And then during my career, I've been moving towards computing bioinformatics and computing biology. And now I am in between all these fields. So I will tell you a little bit later. And the center I'm doing my research now is in Barcelona, and it's called Barcelona Supercomputing Center. And it's a center that has a big machine. It's called Mare Nostrum. And it was built, the whole center was built around this big machine. It's a big computer, supercomputer. And the peculiarity of this supercomputer is that it's within a church. In Barcelona, we needed some space, and we only had a church to put the computer. So this is how the church looked before, and this is how it looks now. Is it really impressive? So if any of you come to Barcelona, send me an email and I'll show you this, and you take some pictures, and it's very nice. But we also use it for other things, of course. So basically, my research group and my research for the past 25 years has been focusing on the relationship between, I'm sorry, I don't know, I cannot get rid of this PowerPoint thing. I'm sorry, I don't know. Okay. So my research is centered in trying to find the relationship between a change in the genome and a phenotype or a disease, basically. Okay, we are interested in several phenotypes, but mostly in diseases. And in the group, we have three different lines, research line, tackling microbiome in the gut, mostly. Now there is a big wave of metagenomic analysis going on directing towards the clinic. Then we have a lot of activity on cancer genomics. I will tell you some examples, and also on the understanding of genetic basis of complex diseases, okay, which have to be treated differently. But the essence is to follow this dogma. I'm sorry if it's very easy for many of you. It's probably very easy for many of you, but I wanted to put you into context, which basically what we care is what happens when there is a change in the genome. There is this change in the genome is transmitted to the RNA, to the protein function. Then we have a misfunction of the protein, and we can have disease. Okay? But this disease can be tackled in different layers of complexity, okay? From the genome itself to our body. There are different layers. You have the genomic information is transformed into metabolic and molecular information, pathways, systems. This is all together is put into a cellular system, then become the cells, the tissues, the body, and one single change, one mutation, one small mutation in one nucleotide will probably affect one RNA, one protein, but then this protein can be involved in several pathways. So the propagation of the error, it's exponential. So It's this protein, then different pathways. These pathways can be affecting different cells. These cell types can be in different tissues. And at the end, the patient goes to the doctor and tells him that that hurts, or he has headaches, or he has, and there are many processes in between, okay, to try to understand what's happening with this patient. The point here is to go to the lower level to see the origin, because most of the text, I don't know whether you can see it, I cannot see it, but down there is the clinical tests. Most of the clinical tests are on that part, okay, are on organ, blood test, morphological aspects of the patient, and most of the times we treat the symptoms only, or secondary symptoms, but we don't know to the origin of the disease. Okay, so it's very important to have this in mind, because our goal is to go to the genomic change. Only on genetic diseases, a flu or an infection, it's different. Okay. So what kind of mutations do we have, or what kind of mutations can we have? We have germline variation. This is the ones that we get from Papa and Mama. Okay, this is our basic machinery. We cannot get rid of that. All the cells have this basic machinery. Then there is another type of mutations, which is de novo mutations. We happen, so Papa and Mama do not have those mutations, but the mutation happen in their germline, okay, in their sexual cells. And these are transmitted to the individual. Here, most of the tissues have the mutation. If it's during the development, some tissues have the mutation, so it's different. And then the last part, or the last type of mutations, which are very important in cancer, are the somatic mutations. Those happening in our cells during our life, and that we do not transmit to our children. Okay? But those are, as I say, very important for cancer. So these mutations, as I said, the genotype, which would be what we acquire from Papa and Mama, is our basic machinery, genetic machinery, and this genetic landscape that we get from Papa and Mama already determines us either to prevent for some diseases. Some people have a very better immune system than others. Okay, some people get cold when it gets a flu, and others are more resistant. We know this. This is because of our variants, probably dealing with the immune system. Other variants give some risk to disease. Are not really. It doesn't mean that if you have the variant, you have the disease. It puts you in a risk situation where, if you then combine with a not very healthy lifestyle, you end up having a disease. Okay, so it's very important to know also these mutations that predispose to disease. And also when the germlines, there are some mutations in germline that we get from Papa and Mama, and if we get them, we get the disease for sure. This is cystic fibrosis and the cysteinurias, many monogenic diseases, what we call. Okay? And the last ones are the somatic mutations, which are caused by chemicals and bad things of the world. Okay, bad things that generate mutations in our genome. Of course, those somatic mutations are happening on top of our basic landscape of our genetic background that we get from Papa and Mama. Okay. So, but the way the mutations affect every of us in terms of disease is very different. So, as I said, in some cases, if you have the mutation, you have the disease. There's no doubt about it. Those types of mutations or variations that we call variations better than mutations are very rare in the population. Okay, few people have it. Other variants that tend to predispose to disease start to be very common in the population. Okay, variants that predispose to cardiovascular disease, diabetes, asthma. These variants are very common in our population. Okay, many of us have those variants, but it's very complex because it depends with which other variant you combine, etc. Okay, but they have low penetrance and they are very common. And normally these variants are affecting regulatory regions in the genome. Okay, the effect is mild, but over the years you start accumulating this defect, and at the end you get the disease. Okay. So our goal and the goal of modern medicine is to try to link, as I said, a variation in the genome with some phenotype. In order to be clinically has a value at the level of patient, you don't need to know actually what this variant is doing, which pathway is affecting, with protein. For diagnosis, as a marker, you don't need to know. You just need to correlate mutation with the disease. It doesn't happen if, it doesn't matter if you don't know in the middle what's going on. Okay? It has a clinical value already. But the point, in order to develop therapies, and to try to, yes, mostly to develop therapies, it is very good to know what are the molecular bases of that mutation. What protein is affecting, which pathway is affecting, and all those things. Okay? So there have been many ways to try to find the gene or the mutation that cause a disease. Traditionally, as I did it in my PhD, traditionally there was an approach that went from the function to the genetics. Okay? In 1999, 2000, when I finished my PhD, most of the people were trying to find disease genes by starting with the function. So we knew, for instance, in my PhD, I was working with amino acid transporters in the kidney and in the gut. And one amino acid transporter fit very well all the physiology described for a particular amino aciduria. Okay, so the patients had, in this case, they had intolerance to some amino acids, they had vomits, a lot of protein in the urine, etc. So by knowing the function of this protein, that was my PhD, and knowing the phenotype, we could guess and we could match these two elements, but at the end we had to go to the genome and demonstrate that that gene that codes for this transporter was really mutated on those patients. Okay? And that was the approach. So from the function to the genetics. Other hypothesis-free approaches were based on linkage disequilibrium analysis, on satellites and pedigrees. It was quite complex. Okay? But now the picture is different. Now we have there's many planets have aligned, okay, and we have a very nice situation now, in which mostly the availability, so the large production and the low price of sequencing, okay, is giving us this opportunity, and very important, the knowledge that we have now on the genome, what we say the genome annotation. We know very well. It's not everything, of course, but we know very well many of the regions in the genome, what they are doing. So it is necessary when you want to try to infer which mutations has what functional implication. Okay? So now we can sequence many patients, we can interpret the genome, so why not do it the other way around? Instead of going from the function to the genetics, let's go the other way around. Let's go from the genetics to the function. Okay? So now we can gather many patients and controls based on the phenotype. For example, type 2 diabetes patients, a cohort, we gather, I don't know, there are many cohorts on, particularly on type 2 diabetes, thousands of patients. You have to have the control, so people without type 2 diabetes. And the point is to sequence or to genotype their genomes, then to try to identify the differences between normal and diseased genome. Then you have to do a functional analysis, so you have to try to infer this mutation, what is causing in our physiology. And at the end, you end up identifying some mutations that are linked and some genes that are linked to the disease. And from there, you can start doing more functional studies. Drug discovery, these genes here become drug targets, okay, for therapies. So this is the basic scheme of the large genetic studies that we have every day, and they are growing and growing and growing. Normally, they are growing in terms of patients, more patients, and I think this has to grow because it grows the statistical power that we have to link mutation and disease, but we have to work also on the intermediate layers to make the whole protocol more efficient. So this is what basically we are involved in our center, in my group, in the Barcelona Supercomputing Center in Barcelona, together with many hospitals of Barcelona, and our role or our position in the whole pipeline would be this one, would be the analysis of the genomes, basically finding the mutation and interpreting what could be the functional consequence of that mutation, to then send it to different places, this information. It depends for what we want. It's either research or directly to treat the patient. Okay, so it could be to treatment development or to diagnosis and in this case, prognosis. So, but depending on the mutation, we can start applying. This is something that we want to apply, and I will tell you at the end, to regular healthcare system, and many hospitals are already applying it, mostly on cancer. But there are different layers of complexity and different ways in which we can apply this information to the clinics. In terms of research, so in order to find mutation and phenotype associations, there are different ways to do it. For germline mutations, what we do is GWAS analysis, genome-wide association studies. Okay? And these germlines, as I said, these germline variants, there are described in the whole human population, I don't know, 15, 20 million of these variants. You have to consider that we have 3,000 million nucleotides in our genome. So a tiny fraction is variant among the population, and the rest is the same. So these changes are making us taller, the nose bigger, more beautiful, more susceptible to fear. I mean, on physiological, on the psychological aspects as well. So, for example, the variant is Mary and Anna. Mary has an A in one position, and Anna has a C. Maybe it means nothing. It has no functional consequence. It could mean that Mary is shorter than Anna. Okay. It could be that even they have different psychology or character. Okay. Could be that Mary with a G here is more protected to disease in general, is more robust than Anna, that would be more prone to disease. And at the bottom, I don't know whether you see it, this is very important. It could mean that one particular treatment works well for Mary, and the same treatment doesn't work for Anna. Okay, so this is the basis of personalized medicine at the level of treatment. And what do we do here? What we do is we gather, I'm sorry, this is misconfigured, but anyway, we gather patients and controls, and here the only thing, the basic thing that we look at is we want to see what is common in the patients at the level of genome that is not common in the controls. Okay, it's as basic as this. There is a huge statistical analysis behind, and it's quite complex, but the basics is like this, and in cancer the same. So you have to imagine that you are gathering thousands of patients, so you want to use your force, okay? So you have to look for recurrency, so things that are happening on the patients that is not happening in the controls. This is the basic thing. In our group, we have been developing, now we work more on bioinformatics, and we have been developing pipelines and tools for this kind of analysis to make it easy, because it's quite complex. Like this guidance, for instance, this is one application that we have done in collaboration with computer scientists in our center. I'll tell you later. And we have been applying this to type 2 diabetes, for example, and other diseases, asthma, etc. And this is just one example of this study that we have done. We have gathered many cohorts of type 2 diabetes, publicly available cohorts, and we have analyzed it with this tool, and we have found several loci, several regions in the genome that are associated with the development of the disease of type 2 diabetes. Okay? And in particular, we are very happy because in particular, we have one variant in X chromosome, and this variant is rare, and the penetrance probably is higher, which is like this because we have an odds ratio of almost 3. This odds ratio means the risk over the average population that you have to get, in this case, type 2 diabetes. This example is very nice. I will not enter into details, but basically we have found one change that was very far from all the genes, but was in one enhancer, and this change is affecting the binding of a transcription factor, and it's affecting the expression of a gene that is related to insulin resistance. Okay, so all this makes the story quite nice, and we are following other functional assays behind with, in association with different doctors and molecular biologists. Using this type of approach in different ways, there have been many variants associated to many diseases. Okay? This is only risk. This predicts only risk. It is very difficult, and we don't know still how we can translate this information into clinical practice yet. Okay? Into what we call personalized medicine, that I will explain to you later a little bit better. This is still, it's very hard. You cannot tell a patient you have 1.3 possibilities more than average to have Alzheimer's, for example, or to have diabetes. The patient doesn't know how to handle this information, so you have to be careful. So on this side, the programs on personalized medicine still are working on this, and we're not considering to apply it right away. The other field that we are working quite a lot, and it is already entering into the plans of personalized medicine at many levels, is cancer genomics. This, again, as I remind you, is most of the cancers are caused by somatic mutations, so mutations that are caused by chemicals during our life, okay, generating mutations in our genome. And this is very important because what we understood, or what we called prostate, or this is a generalization, and some tumors have a more fine definition, but in general terms, patients with prostate cancer are treated more or less the same in general. Okay? But the origin at the level of genome and at the level of protein, which functions are the ones that trigger the tumor, could be very different. Okay? So in this case, different mutations in different parts of the genome could lead to prostate cancer. Okay? So it is very important to understand what is the origin of each one of these tumors to try to adjust the proper treatment. The treatments are not still there, all of them. Some of them are there, but not all of them. Okay? But This is something that we can start applying for some tumors, and of course, all the research behind is going to provide more information, and it's a dynamic process that will end up having the personalized medicine around us. All the cancer genome analysis, we have done it inside a consortium, a big consortium, ICGC, International Cancer Genome Consortium. This is all the countries in the world are involved here. In the States, it's a TCGA, which have been included here as well. And the first goal of all this analysis was to sequence 500 patients of one particular tumor and try to find all the genes and all the variants that could be drivers, so that could trigger the development of a tumor. Okay? The tumor is a very complex process that has many steps and many layers of mutation accumulation, but it is believed that 1, 2, 3, very few mutations at the beginning are the ones that trigger the whole process. Okay? So the commitment was this: every country had to ask for money to their governments to sequence these 500 genomes, and it has been a success. A lot of Nature papers, a lot of new genes. So there has been more advance in cancer, in the genomic part of cancer, in this last 5-10 years than in the previous 20 or 30. Okay, it has been impressive, and in the next years it will be growing exponentially as well. All this consortia is working on a basic protocol, which is here, which could be a protocol very similar to the personalized medicine protocol that we want to establish. We start with getting normal and tumor samples from the patient. We sequence the genome, we identify the mutations, so comparing the normal genome and the tumor genome, just which somatic mutations have the tumor genome acquired, that could be the cause of the tumor. Then we try to interpret that at the functional level, which genes are affected, how could this affect at more metabolic and physiological level or cellular level, and the point is to then identify specific treatment and to return to the patient. Okay, so this cycle is the one that we have used. At the BSc, in my group, we have been contributing in different ways to this project, and we have generated SMAFFIN. It's a new software. Well, it's not that new. We published 2 years ago, but it was within this environment, and we wanted to cover basically the lack of specificity that many of the methods had when trying to analyze the genome. Okay, you can spend millions of euros or dollars trying to sequence patients. You can have everything, but if you don't have a nice and efficient way of extracting the information from the genomes, this is worthless. Okay, it makes no sense. So there were many different methods to analyze the genome, but we generated this SMAFFIN. We have demonstrated that it has a very high specificity. This is what we want, because we want to apply it to the clinics, and applying it to the clinics, it's better not to say when you're not sure, but if you say something, you have to be sure that's the correct answer. So we go for specificity, not so much for sensitivity. And the point was that after we did it, I will not enter into detail, we demonstrate how well it works with mantle cell lymphomas and other types of tumors. But one very important thing is that it was not necessary, it was not enough just doing a software that is able to analyze genome. You have to do a software that can process many genomes at a time, and that can process thousands of genomes. So traditionally, bioinformatics, we just cared about the program, and we didn't care whether it took 1 week to run and whether it used the machine inefficiently. We didn't care about that. We just cared that the result was fine. But here it's not enough, because the production of data is so huge that genomes are literally accumulating in disks, okay, in hard drives. Literally. We have 8 petabytes now in our center, and we have genomes every day. They come new genomes, and the bottleneck actually is the analysis part. And in this case, the SMAFFIN, the software we did, was very efficient at the level of finding the mutation, but was not very efficient at the level of computing resources, how to use computing resources. Actually, we needed, the first version of the program, we needed a 2 terabyte RAM machine to run one single patient. This is, I mean, with this panorama, you cannot think and translated this into the clinics. So what we did was contacting to computer scientists, computer engineers. This is very important because they come into the play for this reason. We managed to parallelize the program, and now we can process, now in the Mare Nostrum, we can process around 100, 200 at a time patients, and we can get the answer in a window of 10 hours. Okay, so this is quite good. This is the summary of SMAFFIN. In one single program, we can identify all types of somatic variation. We get base pair resolution, so this is very nice to infer then the function of the mutation. We have very high specificity. We have high speed, so we can process many patients. These two parts, so that the fact that we identify structural variation and we have base pair resolution, allow us to characterize complex karyotypes that are happening in tumors. And all these possibilities make this as a realistic solution to start applying it into a clinical environment, okay, not only in research. We started applying this, and we are applying this program to many projects, in particular to chronic lymphocytic leukemia consortium. This is a Spanish consortium within the ICGC. And here what we did was analyze 150 whole genomes, okay, and characterize the landscape of structural variation. So all the chromosomal rearrangements happening in the tumor. I will not enter into detail, but basically this chronic lymphocytic leukemia is characterized, and there is a high frequency on one region in chromosome 13, which is deleted, and we think there are some microRNAs there that are necessary for the cell cycle, and this is one of the causes, one of the major causes of triggering chronic lymphocytic leukemia. Also, mutation of Notch, P53, all these normal genes as usual. We also were able to characterize very accurately these karyotypes of this patient. Okay, this is a patient that has 2 catastrophic events that shuffle all the genome. Okay, actually, this is the genome of the tumor. This is how all the combinations of different chromosomes and different parts of chromosomes. Actually, if this had been in my first exam in biology, my first year of biology, and the teacher would ask me, the professor would ask me, could some living cell live with this type of genome? I would say, no, everything has to be perfectly ordered, and well, it can live, it's not only this, it lives, these cells multiply faster than the others, okay? So, and it's very important to know all these changes and all these rearrangements. Of course, all these we have validated using experimental techniques. We are applying also this analysis and this type of analysis to other types of cancer events. I will not enter into detail. And most importantly, we are now entering into a large-scale area, okay, large-scale field. Actually, during 5 years, all these countries that I mentioned have been gathering many genomes. Each country has been gathering many genomes of their specific tumor type. And at some point, the consortium said, okay, we have now, all the countries have a lot of genomes, let's put them together, and then we gain statistical power and do a pan-cancer analysis, okay? In the US, TCGA did a pan-cancer analysis, but based on exomes, so only the exomes of the genome. And this new pan-cancer analysis is based on whole genome. So we also analyze the regions between the genome, so regulatory regions and so on. So the point, we managed to put together 3,000 tumor normal pairs of more than 20 types of tumors. Okay, this is a very nice collection of data. These are some details. And we are very happy because our center, Barcelona Supercomputing Center, is one of the few centers in the world that we are analyzing this data for the whole community. Okay, this is 3,000 tumor normal pairs. Is a lot of, it's 1 petabyte of memory of disk and a lot of CPUs to process that data. Okay, so you need supercomputers or you need centers with a lot of resources. I will not enter into the protocol, it's quite complex, but basically it has to go through different steps. This would be the FASTQ, that's what comes almost directly from the sequencing machine. We have to transform it into what we call a PAM file. PAM file is now the coin of the currency of genomes. We exchange genome in this PAM format, okay, which is more compressed than FASTQ. This PAM format is then used to analyze this genome and to find mutations. And then the point was to give the whole community all the variation, all the mutations found in these 3,000 genomes for them to start doing other types of more functional analysis. All this activity during many years have at the end put us in a situation where we are starting to have many variants that have corresponding action at the level of clinics. So that means that for many tumors, when we find a mutation in a particular gene, we can already prescribe a particular treatment for that patient, okay? It's not for all the tumors, it's not for all the mutations, it's for part of it, but this is the starting of the whole process, okay? So these are some examples of some mutations that related to the treatment, okay? And in our center, what we are starting to do, and in Spain and in Catalonia, our region, we want to gather all this experience that we got during years on germline analysis, on cancer in different projects, okay? And we want to put that together and construct what we call a platform for clinical genomic analysis, okay? Where in the bottom layer you would have the developments, software, hardware. Here there would be mostly bioinformaticians and computer scientists working. We want to provide many different types of analysis, from metagenomics, gene expression as well, RNA analysis, and genomic variation, germline, de novo, somatic, and the applications would be in the top layer. They are very wide. Okay. As I said before, the first application where we are going to use this platform that we are building in our center is cancer. Of course, you have to have all types of support from different aspects, partnerships in cancer genomics. We have very nice collaborations with consortium, with many hospitals. We have collaboration with data producers. This is very important for us. Okay, we have also collaboration with large genomic infrastructure like ICREA, ICREA, and we also are thinking on transferring the technology like SMAFFing to a company or to other types of formats. And at the end, we want to give the patient the extract of what we have been doing in research for these years. Actually, in Catalonia, as I mentioned, there have been now, the Catalan government for the first time has put money to push the instauration of personalized medicine at the level of healthcare system, so that really patients can start benefiting from this. And some 3 hospitals have been are joining, 3 major hospitals in Barcelona, Clinic, Vallebron, and Belviche. Our center will do the analysis, and we have ENAC, genome sequencing center, which is also involved. Okay. So this is the first time that, at least in the States, has been happening, but the problem, and you know better than me, is the policy of the hospitals and how hospitals talk to each other. Actually, in Barcelona is also like this. I think in the States it's even worse. But one of the things that we want is to standardize methods, standardize clinical records. Okay? And this is one of the bottlenecks, because each hospital has been growing and developing their own systems. Some of them are applying already some of this methodology to find mutations in cancer. Other hospitals are not there. I mean, it's very heterogeneous, the situation. And the point and the effort now is to try to construct a common layer, okay, where hospitals can talk to each other and have homogeneous protocols. And also very, very important is my last slide, which is about the different communities involved in here. When you're studying and when you are younger, you are more naive and think, okay, yes, but if a medical doctor wants something and a computer engineer wants the same, they can sit in a room, talk, and get something done. No way. This is not happening. Okay, it's like sitting in the same table, a Chinese that doesn't only speak Chinese, and a Spanish, a guy that only speaks Spanish. They don't understand each other. They talk, and everyone tells their own story and what he wants, and the others say yes, but at the end, nothing happens, and it's right like this. So now, all this scenario of personalized medicine involves this different community: medical doctors, molecular biologists, bioinformaticians, and computer scientists. Okay, computer scientists are the newcomers, because they have been doing things for communication, still apps for mobile, and these kind of things are more. But now they see that biology is a nice field, and we actually need them. So we need to talk to them, to tell them what we want, and to shape what we want a little bit. So they can only talk to us, to bioinformaticians. And and medical doctors and molecular biologists can also talk to us. So we are now becoming a little bit the center of the whole play, because we know other languages. Only because of this. It's not because we are better or worse, it's because we can communicate with people. And this is very important, so I would recommend you that you incorporate opening to other fields, not with activity, but just listen what they want, what type of analysis they do, what their fears, these kind of things. You have to be open, because at some point, if you want to do something, you will have to learn another language, not only English, but also computer science, or more medical, more biomedical, or molecular science. And with this I finish. I just want to give you some take-home messages. Large genetic studies are accelerating the knowledge of genetics in disease. Its application into healthcare system is a reality, and will allow personalizing medical practices in the near future. Okay, and very important, implantation of personalized medicine into healthcare system involve the coordination of different fields of expertise and different communities, which is not easy. And with this, I thank all the people that did the work. I didn't do much; they did it, and other collaborations. So thank you very much, and I'm open to questions.
Большое спасибо. Давайте приступим к вопросам. И вот давайте начнем с молодого человека. Он в прошлый раз хотел задать вопрос, но, к сожалению, не получилось. Друзья, на самом деле, после часа будет дискуссионная площадка, сможете нашим спикерам задать интересующие вас вопросы. А пока вот несколько вопросов на данный момент.
Thank you very much for this very useful information, Dr. Torrents. And I just want to ask you only one question. I'm the PhD of Russian Medical Academy of Postgraduate Education. This is from Moscow. And you investigate the relationship between different types of diseases and different polymorphisms, polymorphisms of genes. Sorry. And we investigate the relationship between different polymorphisms genes and the efficacy and safety of therapy. We have published many of publications in journals of Scopus and Thomson Reuters with not bad impact factors. And if you have maybe some interest to add new branch in your I want to contact you maybe, and this is very interesting for us because you have very good soft, very progressive, and we have interesting to do.
Sure.
Yes, thank you. And very small question, second. Sorry. About statistical analysis, what type of analysis you use to describe their mistakes? Because as I know, the correct of Bonferroni correction doesn't work. Then you make multiple analysis as you do.
Yes, that, the error in general, it's one of the major problems in life and here, and that's why we tend to not to be in the border of making a lot of mistakes and being able to analyze many samples. We are now in a moment where, as I said, we prefer not to say anything if we're not sure, and say something only if you are sure. Most of the methods to correct the errors or to correct multiple sampling, all these typical statistical errors, we apply them, but still, each one of these studies has so many variables, starting from how do you classify patients, for example. Patients, one could think that it's very easy to just gather people with type 2 diabetes. Well, type 2 diabetes. How do you find type 2 diabetes first? Which level of sugar we have, which level of insulin resistance you have to have. So each one of the steps has many possible errors that you try to control. But at the end, what we do is we calibrate the system or the whole pipeline with independent methodology, mostly experimental. For mutations, for example, we calibrate the whole system, the whole pipeline with PCR and Sanger sequencing, okay, which is independent, and other methods. And this is the only way where we can be sure that, okay, every time we say this is a mutation, 99% of the times we are right. But this 1% will always be there, but we have to live with this 1% in other aspects of our life. So it's some error we cannot avoid.
No need to cover this all in Russian. Молодой человек представился и поинтересовался, возможно, ли будет сотрудничество, если я правильно понял, школа после дипломного медицинского образования. Замечательно, вы лучше меня справились. А также поинтересовался, каким образом контролируется наличие потенциальных ошибок на выходе. И Дэвид еще раз повторил, что они предпочитают не гнаться за самой гранью и максимальной чувствительностью и обрабатывать один образец за раз, а также в случае, когда они понимают, что, возможно, есть некая неуверенность, ошибка, просто не говорить ничего и таким образом не давать людям ощущение неуверенности. Естественно, они применяют все существующие методы статистической минимизации ошибок, но также всплывает такая такие тривиальные вопросы, как каким образом категоризовать пациентов. Ну, давайте соберем пациентов с диабетом второго типа. А каким образом мы определяем диабет второго типа, какой уровень сахара? Таким образом, еще им приходится калибровать систему, они, как правило, параллельно со своими предсказаниями пользуются независимыми методами, такими как секвенирование по Сангеру и ПЦР, позволяющими сравнивать результаты их предсказаний с этим независимым методом, таким образом управлять систему, в заключении достигая примерно 99% точности, ну а оставшийся процент с ним приходится жить, но в жизни везде есть такой 1%, с которым нам приходится жить.
Спасибо. Друзья, еще один вопрос. Вот давайте девушку.
Thank you for lecture. I have two questions. First is if your software is freely available. And the second one, could you provide several examples of real translation to clinic? Like you find a variant and you go to doctor and doctor know what to do with this, especially not only cancer-related disease.
If I could translate, sorry. Два вопроса. Первое, в свободном ли доступе это софт? И второй, не могли бы ли вы привести какой-то конкретный пример использования этого софта, что человек приходит с анализом к врачу, и врач знает, что делать, особенно какой-то пример, не связанный с раком?
Yes, I'll try to make it easy for you.
That's the author of the paper you've been reviewing, by the way.
Okay. Yes, the software is free. Unless you want to make money with it, then it's not free.
Если вы хотите на нем зарабатывать, то тогда не бесплатно.
But if it's for good cause, yes. I mean, for academics, it's completely free. Most of the software that it's available, it's free for academics. One thing is that it's free, and the other one is that you are able to use it. Because it's complex, you need expertise, you need machines, so it's not that straightforward. Examples that I know where genetics is being applied to clinics, yes, for many years prenatal genetic studies are these, actually. And on many monogenic diseases, at least for diagnosis, not so much for treatment, but for diagnosis has been working already. For treatment, the field I know most is cancer, and yes, cancer, it's already being applied in many hospitals, in the US, in many hospitals, in Barcelona, in Germany, but as I said, at the level of hospital. Okay, so there is the oncologist, the team of oncologists that gather and analyze the situation. Right now, they base their decision on the previous experience of other hospitals and other people, the research papers and so on. And the whole point now is, which is very important, is to generate a layer above all the countries where we can share information. So treatments that has been a specific mutation that has been found in some patients in Kansas, and some treatment has worked, that this information could be used next day or next week in Barcelona or in Germany or in Kazan. So this is also going to be very difficult. But yes, there are specific examples, BRCA and lung, yes.
Yeah, I mean something new, not cancer, like not prenatal, which we know.
Outside cancer, for treatment, still it's early for that. And on the other part, on which treatment, not only cancer, I mean, whether aspirin is better for some people than for others, this is early for this, but it's going in this direction, but it's early.
Давид ответил, что его софт бесплатен, как и большая часть программ для академиков, она обычно бесплатна, но тут нужно понимать, что конкретно эта программа требует достаточно большой экспертизы для ее использования, а также большое количество вычислительных мощностей, так что, скорее всего, просто так и пользоваться не получится. С точки зрения применения, типичные примеры — это пренатальная диагностика, которая используется уже много лет. Также Давид по большей части знаком с диагностическими методами подбора лечения именно в области рака, потому что текущие исследования сконцентрированы в основном на этом. Как правило, требуется команда специалистов на уровне больницы, которые вместе собираются, принимают какое-то решение, основываясь на опыте с предыдущими пациентами, а также на знаниях, которыми они делятся со своими коллегами из других центров. И Давид еще раз заострил внимание, что очень важно, чтобы эксперты, пользующиеся этими технологиями, делились, и чтобы, если у вас сегодня получился определенный метод лечения, основываясь на определенной диагностике, завтра этим уже мог воспользоваться кто-то другой. С точки зрения применения индивидуализированной медицины, не связанной с раком, с точки зрения терапии, например, понять, кому аспирин поможет больше, а кому меньше, к этому все идет, но пока мы еще до этого не дошли.
Спасибо большое, друзья. На этом вопросы закончим. Можно аплодисментами.